Hi and welcome to this video on the communicable diseases topic, fighting disease with drugs. When a person is infected with a pathogen, they would often look in their medicine cabinet or go to their doctors to get some form of medicine that's going to make them feel better. Sometimes the medicine only treats the symptoms. Examples include decongestants and painkillers. Many of these drugs as well are derived from plants because nature is the best architect for our medicinal arsenal. And one well-known painkiller is aspirin. And not only is aspirin really good at reducing pain, but it also minimises fever. It is anti-inflammatory, so it reduces inflammation or swelling. But it's also really good in preventing heart conditions. So this is derived from willow bark. Another drug that's well known for preventing heart conditions is the drug digitalis. And this drug actually comes from foxgloves. On the other hand, you have drugs that cure the illness. And some of these drugs, in fact a lot of them, come from our fungal friends. Thanks to Alexander Fleming, we have discovered a whole heap of antibiotic drugs. So rumour has it that Alexander Fleming was quite a sloppy scientist and he allowed his bacterial culture to get contaminated with a mould, the same sort of mould that you might see on bread that's gone past its sell-by date. And the mould landed in his dish, which he was culturing the bacteria in, and around the mould there was this zone where no bacteria existed. So he worked out that there must be some chemical in this mould that is killing the bacteria that were growing on his plate. And this chemical was identified and became our first antibiotic, penicillin. The difficulty was extracting enough of this chemical from the mould to be effective en masse, and it was only until Florey Chain, about 10 years later, managed to figure out a way of extracting enough of this chemical and mass producing it so that we could use antibiotics to revolutionise medicine. Problem is now we've got bacteria that have evolved resistance to every single antibiotic that we have. So scientists are working round the clock globally, digging up soil, looking through soil samples to find any more fungal species that could potentially be our next antibiotic weapon. When a new medicine is being developed, and this could be a medicine that's there to treat an illness or actually cure the illness itself, there are certain criteria that have to be met. And to ensure that all of these categories are met, the drug is rigorously trialled. Any drug that is being made has to be safe, so it mustn't be toxic. It cannot poison the person, and there have to be few side effects. Now, there are very few drugs out there that have zero side effects. If you look at the packets of any medicinal drug, you will see that there are side effects such as nausea, migraines, headaches, weight loss, weight gain. All of those side effects, although annoying, they are a small price to pay for the greater good. And looking at the safety of a drug, it's often a case of, are the side effects outweighed by the benefits that the drug will serve? The effectiveness of the drug is important because fundamentally the drug needs to do what it says on the tin. It needs to either treat the symptoms of the illness, cure the illness, or prevent it. So often with preventing illnesses, you're looking at vaccines. And there is another video on these if you wish to view it. A drug also needs to be stable, so it needs to be stored easily. You don't want a drug to spontaneously combust if it's left out in the air. That wouldn't be any good. Equally, if you rummage through your medicine cabinet when you've got a headache, you think, oh, have I got any painkillers? You might find one that's been sitting in there for a couple of years. It will still be okay. Often these medicines are designed to last for years and years and years and still be stable. The reason for this, of course, is that the people developing these drugs would have to spend a lot of money, millions and millions and millions of pounds, to develop them. This whole drug trialling process can take up to 20 years. So if they're going to make a drug that goes off in like 3 days, what's the point? But also, can it be used under normal conditions? So it doesn't need to be taken under high pressure or anything like that. It doesn't always have to be administered in a hospital by a trained person, etc., etc. Can it be successfully taken into and out of the body? So it needs to, whichever way it gets into the body, whether it's taken orally in a tablet or it's injected, bearing in mind if a drug needs to be injected, that should often be done by a trained individual, or certainly a person would need to receive training who would be taking that drug on a regular basis. But it needs to reach its target. So if you've got a problem with your lower digestive tract and you take a drug orally, is it going to get past the barriers that are in the stomach, or is it going to degrade in the stomach and not reach its target further down the line? So that's really important. Equally, once it's done its job, can it be safely removed from the body? So what normally happens with drugs is that they often get broken down by the liver into safer products and then excreted from the body. If it can't do that, it can't pass its test. When scientists are trying to develop a new drug, the first thing that they tend to do is look for where that illness that they're developing the drug for has originated, because it's very likely that there is a plant from that region that might have some chemical that could be put to trial. They will look at the chemical components of such plants and they will input that into a computer simulator model, and they'll try that simulation out on the actual problems caused by the illness. And if the computer gives its version of a thumbs up, then we go into this section of the preclinical trials. And preclinical basically means before humans are the guinea pigs. So the drug in question will be tested on cells, tissues and organs. First of all, is it safe? You can quickly find out if something is toxic or not by the effect it's going to have on these cells, tissues and organs. It seems to be working at this stage of the trial. It is worth noting that a drug could work in a very different way on an isolated organ in comparison to working in a whole living organism. So this is why it is necessary to have an animal testing stage, which obviously is very controversial ethically. But here you can test to see if the drug actually works. Is it effective? Safety is also being tested here and looking for the optimal dose. And once you can work out the optimal dose for an animal such as a mouse, you can then scale it up to a human being later. If the drug passes these steps, then it will go into the clinical trial phase, which involves humans. First of all, a group of healthy volunteers are used. So the small group of healthy volunteers are not trying to find out if the drug is effective because they don't have the condition, but really we are looking at the safety, and if it seems to be safe with very limited side effects, then the dosage can be worked out as well. If the results are good, then the drug gets tested to again a small group of individuals, but this time they are suffering with the illness, and here we are looking to see if the drug is actually going to work. Is it effective? At this particular moment in time, the drug trial becomes very, very scientific. So this group of volunteers gets split into two. First of all, we have a control group, and these should have equal numbers of people, and the control group are given a dummy drug or a placebo. So if the drug that is being trialled is actually taken as a pill, the placebo will be a sugar-coated pill. If the drug in question is an injection, then the placebo would be an injection just of saline water. And the reason why there is a control group of people is because there is this thing called the placebo effect, where somebody who believes that they are receiving a medicine will psychologically start to feel better and will have fewer symptoms. But that is not correlated to whether the drug is working or not. That's just the way the mind works. The data from this control group will be compared to a group receiving the actual drug itself. And again, this can be split into something called a blind trial. Here, the patient doesn't know whether they are in the control group or the group where they're actually testing the drug itself. But the person who's collecting the data does know which group they belong to. The problem with a blind trial is that the person could actually have some bias who is collecting the data, and the data might not be a true reflection of what's actually happening. A more scientific way of doing this to prevent that bias is to have what's called a double blind trial. And that basically means that the person administering the drug or the placebo doesn't know which one is which, and the person who is collecting data, it might be the same person, again doesn't know which group the patient belongs to. So it eliminates bias, and it's a much better way of doing science. It's also worth mentioning here that the pharmaceutical company should really outsource the drug trial to an independent company, and an independent company are not being paid by the pharmaceutical company. So they will not receive money from the pharmaceutical company, and therefore they will have no bias. Because what you don't want to do is to have a drug that's been trialled by a company that is paid by the pharmaceutical company. If they're receiving data that says the drug is not working at all, it's got quite a few horrible side effects, that company will rely on the money, and therefore they potentially could start lying about their data, and you don't want that at all. So it's much better to have a double-blind trial done by an independent company here. And if all is going well, then it all gets upscaled. So a larger group of volunteers suffering from the illness. And this is where you kind of get your long-term effects. In conjunction with it being licensed, so that GPs, general practitioners, doctors could actually administer this, every single time a patient that's on this newly licensed drug goes back for a checkup, the data that the GP is collecting goes back to the scientists that are studying this drug to see what the long-term effects are. So to quickly summarise, there are a few terms that you need to be familiar with here. Antibiotics are drugs that actually cure the infections by killing or preventing the growth of bacteria. They do not work on viruses. Antiseptics sound a bit like antibiotics, so you kind of got to appreciate there is a difference here. Antiseptics are chemicals that they also kill bacteria. They can kill viruses as well, but on the surface of the skin. They're a bit stronger than antibiotics, so you cannot actually take these orally or take them inside the body because they'll be too potent. Even stronger still are disinfectants, and these will kill bacteria, viruses, etc. But on surfaces, work surfaces. Disinfectants you don't want to put on your skin. Painkillers are drugs that can treat the symptoms of the illness, they do not cure it. Clinical trials occur when the drugs are conducted on human volunteers. A placebo is a pretend drug that only has psychological effects and is taken by a control group. A blind trial occurs when the patient is unaware who has been given the drug or the placebo, and a double-blind trial occurs when both the patient and the doctor or the scientist or the researcher are unaware who has been given the drug or the placebo, and this greatly reduces any bias. In terms of exams, you could be asked to resequence a mixed up order of the drug trialling process. Logic is your friend here. Or often you might have a flow diagram of the drug trialling process, and there might be some gaps that you need to put in. So do learn the sequence of the drug trial process. That would help you greatly.